All right, so in this video, we're going to talk a little bit about protein synthesis and the idea of what is this DNA all about and then how can we use it. So you guys probably all know that DNA, and here's a picture of DNA here, is, we know these things. We know it's double-stranded. We've seen this. And it's made out of the deoxyribose, and its bases are A, T, C, and G. Okay, so DNA is found in the nucleus of the cell, and you guys always say it contains all the genetic information and all this kind of stuff. So what is a gene, and how does a gene get expressed, or how does, like, the fact that you have sickle cell anemia in your DNA, what does that mean, and what does that look like? And so that's kind of what we're going to get into here. And so the first part is we're just going to kind of meet some of the guys that are involved, so one of the guys that are involved in this idea of protein synthesis is DNA. Another one that's made through transcription is mRNA. And mRNA is this little piece right here. So mRNA stands for messenger RNA. It's carrying a message. And its message is the gene. Messenger RNA. I spelled messenger wrong. And messenger RNA, as most RNAs, are actually single-stranded. And in the case of RNA, the sugar in this case is actually ribose instead of deoxyribose, as in DNA. It's kind of part of why it's single-stranded. Its bases, instead of being A, T, C, and G, are A, U, C, and G. So there's no T. And what it does is it's going to, messenger RNA particularly, is going to carry the code for making proteins. From DNA in the nucleus because remember, the nucleus, you want to protect your DNA. So you have to have a way for that information to go out from the nucleus out into the cytoplasm. So from the DNA in the nucleus to the ribosomes, which are in the cytoplasm. And so let's actually meet a ribosome next. So what is a ribosome? Ribosomes, here they are. This is actually a picture of one coming together. So this is actually the small subunit, and then the small subunit kind of slides into the large subunit, and so you end up with one ribosome. And these guys are also usually made through transcription. Mostly through transcription, because they're about 60% ribosomal RNA. They have their own RNA. So that would be made through transcription. And then they're 40% protein, which is actually made through this process that we're going to talk about, protein synthesis. So it's transcription and translation. And this is where protein synthesis happens. Okay, so this is the site of protein synthesis. And then the last kind of guy that's involved, or molecule that's involved, is also made through transcription. And it's this friend. This is tRNA, or also known as a transfer RNA. And it's got some different parts to it. This part right here is where the amino acid would attach. So there's 20 different amino acids. So whatever amino acid this one codes for would attach. And then right here, these three bases right in here are what we call the anticodon. And they're going to be specific to an amino acid. Okay. And so this tRNA is also known as a transfer RNA, and it carries the amino acid to the ribosome, and it's very specific. Okay, so it's specific to that. And so now we kind of know what all the people are. Let's kind of look at this genetic code and what this genetic code kind of codes for. So within a genetic code, you have what's called codons, okay? And so one codon is actually just three bases in a row. And one codon codes for one amino acid. And we usually abbreviate, we get lazy, and so we just abbreviate those AA. Okay. And so there are actually, we know this, there are 4 DNA bases, right? A's, T's, and C's, and G's. And so there are actually 4 to the third, or 64 possible codons. But there are only 20 amino acids. So what we say about this genetic code is that the code is redundant. So what we mean by that is two or more codons can code for the same amino acid. So like AUC and AUU will code for the exact same amino acid. Okay, two or more codons codes for same amino acid, but it's not ambiguous. Okay, what we mean by that is that no codon codes for more than one amino acid. Okay, so AUC is only going to code for one amino acid. It's not going to code for 4 different amino acids. So here's kind of the big overall picture. And I'll probably be writing this on the board like 1,700 times. Okay, here's our DNA up here. And this is actually what we call the coding strand. We'll talk about how you know it's a coding strand in a second. And then, it's really just because of where the start codon is. Then you have transcription, which then produces this mRNA product. Okay, that's what this is here. And then each of those 3 codons, here's a codon, here's a codon, here's a codon, and here's a codon, will be translated. So translation will occur. And we'll turn it into a protein. So this is an amino acid. We could do amino acid, a different amino acid, a different amino acid, and a different amino acid, okay, based on the code up here. And so that's where you get the protein. And then the protein can go into the cell and do thousands and thousands of regular protein things. It could be an enzyme, it could be a receptor, it could be an activator, it could be a hormone, it could be whatever. It could be a protein. Okay, so let's look and see how this process of proteins are actually made inside the cell. We're going to start with the process of transcription. Transcription is one of my favorite processes because it's so easy. You guys are going to be like so excited. Okay, are you ready? Okay, we have a DNA strand here. Ready? Okay, so what's happened is you can see that it's been pulled apart, so it's been separated. And there's a molecule right here, okay? That molecule is known as RNA polymerase. It's going to make RNA, just like DNA polymerase in replication made DNA. But RNA polymerase is so much cooler than DNA polymerase because it can actually kind of do the jobs of like practically everybody that we met in replication. What it's going to do is it's going to bind to what we call the promoter, and that's how you know the coding strand. And the promoter is also known as what we call the Tata box. I know, you can all giggle. And it's because it's got a TA sequence there. And then it separates the DNA strands. So it kind of does that job of helicase here. And then what it does is it base pairs. So it pairs like here you can see a G with a C, a T with an A, a G with a C, whatever. It just base pairs. So it pairs RNA nucleotides with the DNA bases. And just like in replication, these guys come in as with their triphosphates, and that's the energy that's used to make that bond, that covalent bond between the one nucleotide and the next. And it stops when it reaches a terminator region. There's a few different kinds of terminators, so we don't actually have to know. We just know there's a terminator. Reaches terminator. And here's our little mRNA coming off. mRNA. Now, the mRNA, it grows in the 5' to 3' direction, okay? So that's kind of how it grows and it's made. And then now what's going to happen to our RNA, okay, so now we've made our RNA right by our DNA, and what we're going to do is we have to process it. Now, this is something that happens only in eukaryotes. It's kind of a key part. I'm probably going to say it about 3 times. So this is RNA processing. Oh, the dog's barking. What a fun treat for you. Only in eukaryotes, okay? Only in eukaryotes. Bacteria don't do this. And that's going to become important, actually, when we talk about some of the genetic variations in biotechnology and stuff. I'm going to kind of color code my little friend here, and I'm going to color code these guys right here. Ready? So that one's going to be red, and that one's going to be red, and that one's going to be red. And then my other ones are actually going to be blue. So this is my mRNA transcript, that thing that I made up here with RNA polymerase. So this is just my little piece of mRNA. And what's going to happen in RNA processing is my introns are going to be removed. And so that's what this piece is. They actually loop up just like this. There's actually some coding right here and some coding right here that causes some enzymes called spliceosomes to loop them up to recognize them. And it literally cuts them out, as you can see in this picture. Okay, it literally cuts these introns out. And what's left over is the part that I'm going to color in red, which is called exons. Okay, and so the exons are then linked together. And I always remember it this way. Exons are expressed. So that's how you know what stays in. The other thing, that's all done, by the way, that's all done by spliceosomes. They cut out introns and put exons together. And the way that they can actually recognize that is actually because they are made of protein and snRNA. It's just spliceosome RNA. Okay, so they have a special RNA in there, so they actually can recognize those exons and introns. There's actually like a little code right there at this region that kind of allows them to recognize it. So there you are. Okay, now the other thing that happens in this RNA processing, so we get out the introns and we keep the exons. There's one more thing that happens, actually two more things. One is they actually add what's called a 5' cap. Now they drew this ginormously. It's actually just on the 5' end, they actually add a modified guanine. Okay, so this is just another part of this RNA processing. We add a 5' cap is added. And they add what's called a poly A tail. And these are really disproportionately because this is 1 G. This is like anywhere between 50 and 250 A's, and it's much tinier, so it's kind of a bad picture that way. So we're going to add a big poly A tail. Both of those help target and they actually help prevent the breakdown of the RNA transcript, and so that it kind of helps preserve the RNA inside the cell, so it actually will be able to be translated once it gets outside the nucleus and all that kind of stuff. So because there's actually enzymes that are trying to break down foreign DNA and DNA that maybe isn't being used anymore. So these guys help protect that breakdown. This also, the poly A tail kind of helps with translation too, so that's why they're added. Okay, so just again, RNA processing, 3 things happen. 1, we remove our introns and we keep our exons. 2, we put a 5' cap on. And 3, we put a poly A tail on. Okay, and remember those exons only occur, and the exons and introns only occur in eukaryotic cells. It's